Okay, so we're going to start chapter 4. I've broken this chapter up into 4 parts, and this is part 1. We're going to be looking at cell structure. So to start, I want to talk to you a little bit about cells and the different types of cells and different types of organisms on our planet. So cells are going to be the basic unit of life. Okay, and so what that means is that single cells can actually live, where if we were to take apart a cell, right, which we're going to learn about the organelles, the parts and pieces of a cell, those are not considered to be alive. The cell is going to be the smallest unit that's going to be considered to be a living thing. Okay, so a nucleus is not considered alive. A mitochondria by itself is not considered alive. So again, the cell is going to be the basic unit and structure of life. Okay, and so I just want to briefly talk about prokaryotic versus eukaryotic cells, and we've already really talked about this, so you should have a good foundation. We're going to talk about this more throughout this chapter, but prokaryotic, pro means before and kary means nucleus. So prokaryotic organisms are going to lack a membrane-bound nucleus and other membrane-bound organelles. Prokaryotic organisms are also single-celled. Okay, so when we see the term prokaryotic, we're going to think no nucleus, no membrane-bound organelles, and all organisms that are considered to be prokaryotic are going to be single-celled organisms. And we're going to learn that they belong to 2 domains, the domain bacteria and the domain archaea, or archaea. Some people say it that way. The opposite here are eukaryotic cells, and so eukaryotic cells are going to have membrane-bound organelles, including the nucleus. And for eukaryotes, it's a little bit more tricky. Eukaryotes belong to the domain eukarya. They can be single-celled or they can be multicellular. It depends on what organism we're looking at. So for example, all animals are multicellular. Okay, but if we look at, for example, fungi, some fungi are multicellular, and then there are some that are single-celled. So we can't use that as a descriptive characteristic. In these figures, I just have a couple of cells to show you. So here we've got a couple of different bacterial cells. Okay, so again, single-celled prokaryotic. And in this example here, we've got a multicellular. This looks just like the onion root tip from lab, right? So this might be a plant. You can see all of the nuclei here, and you can see their kind of rigid rectangular shape. And then here we have a tissue sample taken from an animal. I forget exactly what tissue this is, but it's a tissue that produces mucus. You can actually see these structures in here. Okay, so again, eukaryotic cells here, eukaryotic cells here. We can see membrane-bound structures, and then prokaryotic cells here. So those are our 2 main cell types on the planet. Okay, how do we see cells? So most cells, with the exception of just a few, are going to be microscopic. They're tiny. We cannot see them with the unaided eye. And so we're going to use an instrument, right, to expand our sense of sight, or to enhance our sense of sight. And so the microscope, right, you've seen this in the lab already. The microscope is going to be an instrument that we use to magnify an object. And so to magnify means to make appear larger. Okay, the light microscopes that we have in class, we just look through the eyepiece, although there are some microscopes where you can actually attach a camera and you can end up with a micrograph. And so all the images that I'm showing you in this PowerPoint of actual cells have been taken, a photograph has been taken either on a light microscope or an electron microscope. We end up with a micrograph. That's a photograph taken with a microscope. Okay, and some of those images are beautiful. The light microscope, which we have here, right, this is the one that we've used in the lab. It's called the compound light microscope, and hopefully you know that's because there are 2 lenses, the ocular and the objective lens between your eye, where you would look through, and the specimen, which sits down here on the stage. And so that's why we call it a compound light microscope. It's light because we're using light down here. Here's our light source. And this light is going to be of the visible light spectrum. Okay, it comes up through the condenser lens, onto your specimen, through the objective lens, through the ocular, and eventually that light is gathered by your eye. Remember that there are 4 different objective lenses. The 4, the scanning lens, the 10, which is low power, the 40x, which is high dry, and then remember our scopes have the capacity to go up to 100x, but that last objective lens here requires immersion oil. So we don't use that in the lab, but just be aware that it's there. I asked you in lab to calculate total magnification. You should know how to do that. That's going to be the magnification of the ocular lens, which is always 10, times whichever objective lens you have in place. So that could be the 40, the 10, sorry, the 4, the 10, the 40, or it could be the 100 if we're using that. Okay. One of the really neat things about light microscopy, which makes it different from other types of microscopy, is that you can view living organisms and you can use special stains, right, to see things inside of those living organisms. You should know what it means to magnify. So again, that means to make appear larger. And you should also know what resolving power means. Remember, that's the ability to see 2 distinct objects as distinct, or we can say the ability to see fine detail. Okay, and the light microscope will have a limit to that. There will be a point at which you can't magnify any farther because you can't resolve. Okay. Second type of microscopy that we're going to talk about, and I want to tell you there's many different types of microscopy. In fact, there's many different types of light microscopy. There's confocal light microscopy, fluorescence light microscopy, there's dark field microscopy, all using a basic light microscope. But I just wanted to touch on really 2 main types. So you got light microscopy, and here's kind of an older microscope. We still have these in the lab, but you guys have been using newer ones. And on the right here, look at this big, you can see there's some people here, right? Look how big this instrument is. This is called an electron microscope. We don't have one of these at MVCC. I used one in graduate school. They're large, they're expensive to run, expensive to maintain. So, we don't need them at MV, but they're pretty neat instruments. So an electron microscope, instead of using light, it uses a beam of electrons. One of the downfalls of electron microscopy is you can't view living things. You have to kill whatever you're going to be looking at. It gets killed in the process if you don't kill it beforehand. So specimens cannot be live on the electron microscope. The benefit of the electron microscope is much higher magnification, much higher resolution, and you can see more detail. We say that you can see ultrastructure. So with the light microscope, you can see where the nucleus is, but with the electron microscope, you can see the detail of the nucleus or other organelle. Okay. So again, high magnification, high resolution. There are 2 types of electron microscopy. There is scanning, which we call SEM, and there's transmission, which we call TEM. And I'm going to show you images produced from the 2 in just a second. But just so that you know, scanning electron microscopy, when we do this, we're really just looking at the surface of the organism. And so the electron microscope, you'll see a 3-dimensional image, right, of that surface of whatever it is that you're trying to look at, the cell or the organism. With transmission electron microscopy, it's a little different. In TEM, you have to actually make ultra-thin sections of whatever you're trying to look at. And then you can label specific structures with electron-dense material. And then when you put that in your TEM, your transmission electron microscope, what you end up with is an image of the inside of the organism. So again, scanning will be a 3-dimension of kind of the outside of the organism, and TEM is going to give you a 2-dimensional image of, but you're going to see the inside ultrastructure of the organism or the cell. Okay, I wanted to just show you the difference between a micrograph taken with a light microscope versus an electron microscope. And so it's the same tissue. And so here we have this tissue and all these little purple dots here. This is an infiltrate of bacteria. And you can see that they're there, but they're teeny tiny. You don't really see any structure. And then if you look here, look at that. This is beautiful. So you can see the tissue that the bacteria are invading, but you can also see these nice rod-shaped bacteria with their long flagellum or extensions coming off their body. Here's another one here actually getting into this tissue. Here's another one. So you can see the difference in magnification and resolution. It's pretty striking. And then I wanted to show you this as well. This is the basic light microscope, right? And then here is a transmission, right? You end up with a two-dimensional, you can see the inside of whatever you're looking at. And then scanning, you end up with kind of a three-dimensional image where you're looking really at the surface. And so what are we looking at? So in all three of these figures, we're looking at this organism here, which is a paramecium. Okay, so it's a single-celled eukaryotic protist. And the light microscope does a pretty good job, right? We can see the plasma membrane or the cell membrane, right? We can see some other structures in here, really prominent. This here and here, those are the contractile vacuoles. Those are going to be used to pump water out so that this guy can maintain fluid balance. And that's pretty much all we see, right? This might be the food gullet here, okay? But we don't see ultrastructure. With the transmission electron microscope now, look at this is the cytoplasm. We could see all kinds of organelles, okay? And then this is probably my favorite picture. This is the scanning electron microscope. So this is the surface of this guy. And so look, it's just coated in cilia, and then you can actually see this little indentation here, which again is probably the food gullet. So you can just see higher magnification, and you can see ultrastructure. Okay, so microscopes are wonderful. They tell us, we get to see the organism or see the cell, and we get to see structures, right? So we can figure out location of things in the cell. We can even, if we're using light microscopy, we can watch things move around in the cell. But what microscopy doesn't tell us is anything biochemical about the cell. And so if you want to learn more about the biochemistry of the membrane, what is it made of? You can't do that with plain old microscopy. You have to actually separate out the parts and pieces of the cell and then study them biochemically individually. And so we've developed a protocol that's pretty easy to do that. First, you do what's called cellular fractionation, which is where you just kind of crack the cell open, dump its contents, and then you do something called differential centrifugation. This is where you actually separate out the parts and pieces. Okay, so what you're going to do here, I'm going to go on the bottom. I will also talk about the top. So we have cells. Okay, and so we want to learn, about the plasma membrane. And so what we're going to do is we're going to take these cells, and we're going to put them in a test tube with some buffered solution, probably that contains some kind of a detergent. And so what's going to happen is we're going to break the membrane, and we're going to end up with all of the organelles, all of the cellular contents inside the tube. So now you just took your whole cell and you broke it open, and you've got all the parts and pieces. So the next thing is you got to start to separate out the components. So this is where you use centrifugation. So there's an instrument called a centrifuge, which will allow us to spin samples at different speeds for different times, and it allows us to separate out the components. So for example, if I take my test tube and put it in the centrifuge and spin it at 600 Gs for 10 minutes, what I'm going to get down here at the bottom of the tube will be the nucleus. So that's how I separate out the nucleus. I can then take all this liquid on top containing all the other organelles, this is called the supernatant, put it into a separate tube, and now I'm left with just the nucleus. I can study the nucleus on its own. If I take this supernatant in another tube and spin it again faster, so for 20,000 Gs for 30 minutes, now I'm going to collect at the bottom of my tube all of the mitochondria and chloroplasts. So again, I can take all this liquid, supernatant off, put it into a new tube, and I can study just the mitochondria. If I take this in a new tube and spin it again in this machine, much faster, so 100,000 Gs for 90 minutes, what I'm going to pellet here is pretty much everything else. The ER, the Golgi, the plasma membrane. Okay, so I can separate out the component pieces of the cell, and I can study those individually by doing cellular fractionation, breaking open the cell, and then differential centrifugation, spinning at different speeds for different lengths of time. When I'm left with this pellet here, the ER, the Golgi, and the plasma membrane, which we're going to learn about, these are all made of membranes. And so I can actually separate those pieces out on what's called a density gradient. And so this is a sucrose density gradient. The least dense will end up here, and the most dense end up down here. So if I put this pellet in there, and again, I spin it for 100,000 Gs, okay, it doesn't say the time, for whatever the time is, I will end up separating out those components based on density. So the plasma membrane is the least dense. It's going to come out up here towards the top. The Golgi is more dense, and the endoplasmic reticulum is denser. Okay, so again, this is a way that we can separate out the parts and pieces and actually study the parts and pieces of a cell. So the microscope is great to look and find stuff, but this is better for studying individual parts and pieces, biochemically. Okay, so let's talk about cells. I want to make sure that you know what the cell theory is. And so way back in 1665, there was a man by the name of Robert Hooke who, using kind of a primitive microscope, he coined the term cell. He was the first person to see cells. And he was actually looking at cork, and he coined the term cell because he thought that what he was seeing under his microscope looked like the little rooms that monks lived in. So that's where the term cell actually came from. And then in the 1830s, there was three other scientists, Schleiden, Schwann, and Virchow, who worked together to come up with the cell theory. And so there's three parts to that cell theory that I want you to know. So first of all living things are composed of one or more cells. Secondly, the cell is going to be the basic structural and functional unit of life. And thirdly, new cells only arise from pre-existing cells through cell division. So there is no such thing as spontaneous generation. Pictured here, I've got some cells that you might be familiar with. These are muscle fibers. Here we've got some red blood cells. And here we've got some compact bone. And you can see some cells in there as well. And so I want to go over a list of characteristics that are shared by all cells. So if we're talking about prokaryotic cells or eukaryotic cells, these are some commonalities. So all cells have a plasma membrane. All cells have cytoplasm. All cells have DNA. All cells have ribosomes. And I'm going to actually add something to the list. So all cells also have some form of a cytoskeleton. Okay. The cytoskeleton is different if we're looking at prokaryotes and eukaryotes, but they all do have some form of a cytoskeleton. So I want to just take a few minutes and focus on prokaryotic cells, and then the rest of this chapter is really going to focus on eukaryotic cells. And so pictured here is the basic structure of like a bacteria. So prokaryotic, pro means before and kary means nucleus. And so these cells, again, will lack a membrane-bound nucleus. However, they do have DNA. I want to make sure that you guys understand that. So if we look here in this picture, you can see it labeled chromosome, DNA, right here, this brown coiled up stuff. This is in a region of the cell called the nucleoid region. There is no membrane around it, so it's not a nucleus. Okay, and just to give you a little bit more information, prokaryotic DNA is circular, and they usually only have one circular DNA molecule. We'll talk more about bacterial chromosomes a little bit later in the semester. So the two domains that have these types of cells are the domain archaea or archaea and bacteria. And again, all members of these two domains, all species, are going to be single-celled organisms. Skip a slide. Just a little bit more information about prokaryotic cells. So again, they're relatively simple, unicellular. They don't have a nucleus. They do have a nucleoid region. They don't have any other membrane-bound organelles. So we're not going to see structures like the ER or the Golgi or lysosomes or peroxisomes or mitochondria or chloroplasts. Those do not exist in prokaryotic cells. However, they do usually have some of these structures. You know what? Not listed here. It was listed a few slides ago. They do have ribosomes. I want to make sure you know that. Okay. They have a cell membrane or a plasma membrane. Some will have a cell wall. The cell wall in bacteria is made up of peptidoglycan, so a little bit different than what we see in eukaryotes. And that cell wall is for protection, for maintenance of shape, to prevent the organism from desiccation, which means drying out. Some will have a capsule made up of polysaccharides, so carbohydrates, which helps the organism attach to surfaces. Some will have flagella, so you can see the single flagellum. Some will have more than one. This is a whip-like tail that's used in movement. Some will have pili. Pili are going to have actually multiple functions. They will help in attachment, adherence to surfaces or other organisms. But most importantly, pili are involved in exchange of genetic information during conjugation. And again, that's a process that we'll briefly talk about in a later chapter. And then fimbriae. Some will have these in addition to pili. And again, these just specialize in attachment to other cells. So all of these, they look like hair-like structures. They are not cilia. They're either going to be pili or fimbriae. Okay, and then I just wanted to spend a few minutes talking about cell size. So whether we're talking about prokaryotes or eukaryotes, cells are relatively small usually. Usually we need a microscope to see them. However, eukaryotic cells are a bit larger, and that's because eukaryotes have organelles that compartmentalize things and make it easier for cells with surface to volume ratio. Okay, so on average, prokaryotes, which are shown here, are going to be about 0.1 to 5 micrometers in size, so pretty small. Where animal plant or other eukaryotic cells, so here we go, are on average 10 to 100 micrometers in size. Now, usually, however, the human egg is actually a little bit larger. The frog egg is a millimeter. Chicken egg you can see with your unaided eye, right? This is just one cell. Same thing with an ostrich egg. So there are some cells that fall outside of that realm, but for the most part, most eukaryotic cells are in the range of 10 to 100 micrometer. Okay, and again, eukaryotic cells are able to be larger than prokaryotes because of the organelles, because of the compartmentalization inside. So why are cells relatively small, especially prokaryotic cells? And that's because cells have to maintain a high enough surface to volume ratio in order for the cell to maintain homeostasis. The cell has to be able to bring in nutrients and get rid of waste products relatively quickly. And if the cell becomes too large, it's difficult to transport, through that vast cytoplasm. And so staying small is very, very important. And so I just want to give you a relative idea here. So these are two different cells, right? They have two different surface to volume ratios. And so this smaller cell has a high surface to volume ratio. So it's got enough membrane, right, where it can bring stuff in and get rid of stuff pretty quickly. As a cell gets larger, what happens is its surface to volume ratio decreases. And there's a point at which the cell, if it gets too large and that surface to volume ratio is too low, the cell will either need to divide to become two smaller cells, or the cell will eventually die. It won't be able to maintain homeostasis. Okay, so being relatively small is important. And again, eukaryotic cells are able to get a little bit larger because of the compartmentalization that we have, because of organelles. So that's it for part 1.